Everyone, welcome back to my YouTube channel. Today our topic is beta oxidation of fatty acids. So first of all, we will discuss what is beta oxidation. So beta oxidation is simply oxidation of fatty acid at their beta carbons, right? It is beta oxidation of fatty acids. Then beta oxidation of fatty acid includes three steps, out of which one is activation, and the second one is the transportation, and the third one is main mechanism inside mitochondria. So first of all, we will discuss the position of occurrence, the way of beta oxidation of fatty acids takes place, and why there is a need of activation. So beta oxidation takes place in cytosol and also in the mitochondria. This portion is the cytosol. The palmitic acid is present in the cytosol, but in order to get into the mitochondria, it's not possible for palmitic acid to directly enter into the mitochondria. The membrane of mitochondria is semi-permeable and it does not allow the entry of palmitic acid into it directly. So there is a need to activate this palmitic acid and to convert it into another compound which named as palmitoyl coenzyme A. This process is actually known as activation, the process in which we convert the conversion of palmitic acid into palmitoyl coenzyme A is known as activation of fatty acid. Like, let's see, here we have equation for the activation of palmitic acid or the fatty acid. See, we have this palmitic acid, and as we want to convert into palmitoyl CoA, so first of all we need a CoA, and this whole process require energy in the form of ATP, so we are also using ATP there. Then this palmitic acid in presence of ATP reacts with CoA, which means coenzyme A, and converts into palmitoyl CoA. Now this palmitoyl CoA is an activated form of palmitic acid, and this palmitoyl CoA can enter into the mitochondria with the help of another enzyme which names crenitine. And this ATP is converted into adenosine monophosphate. This adenosine triphosphate means that ATP having 3-phosphate converted into adenosine monophosphate, which means that there is only one phosphate is present. These rest of the 2 phosphates are there. 2 phosphates that are present here that make the reaction irreversible, which means that now this palmitoyl CoA, activated palmitoyl CoA, cannot be converted back into the palmitic acid, right? So you see in the diagram, here we have palmitic acid that was present in their inactivated form. Now it is converted into palmitoyl coenzyme A here. Having the next step, that is the transportation. The palmitic acid, this palmitic acid that was present in the cytosol in their inactive form has been converted into their activated form, that is palmitoyl coenzyme A, and with the help of another enzyme crenitine, it is transported from cytosol to the mitochondria, inside the mitochondria. Now here the main mechanism of beta oxidation will take place inside the mitochondrial matrix. So let's now discuss the main mechanism of beta oxidation inside mitochondria in the form of flowchart. Here we are having complete flowchart of beta oxidation of fatty acid. If you want to take a screenshot, you can take now. As we have discussed earlier, this palmitic acid that were present in the cytosol converted into their active form, palmitoyl CoA, and then transported into the mitochondrial membrane. And this conversion of palmitic acid into palmitoyl CoA is done under the action of an enzyme named as fatty acyl thiokinase, right? Then this palmitoyl coenzyme A is converted into alpha beta unsaturated palmitoyl CoA. This conversion of palmitoyl CoA into alpha beta unsaturated palmitoyl CoA is done by an enzyme known as fatty acyl CoA dehydrogenase. And during this conversion, there is another product named as FADH2 is also produced. This FADH2 is high energy product and it helps to synthesize ATP during citric acid cycle, right? Then we have this alpha beta unsaturated palmitoyl CoA is converted into beta hydroxy palmitoyl CoA, and this conversion is done under the action of an enzyme named as crotonase, right? Then this beta hydroxy palmitoyl CoA is converted into beta keto palmitoyl CoA, and it is done under the action of an enzyme named as beta hydroxy palmitoyl dehydrogenase. And during this conversion, another high energy compound is produced, that is NADH2. This NADH2 also helps in the production of ATP in mitochondria. Then this beta keto palmitoyl CoA is converted into myristoyl CoA and acetyl CoA, and this is done under the action of enzyme beta keto thiolase, right? Now we will discuss the number of carbons include. So this palmitic acid is 16 carbon, palmitoyl CoA is also 16 carbon, alpha beta unsaturated palmitoyl is also 16 carbon. But this myristoyl CoA is 14 carbon compound and acetyl CoA is 2 carbon compound. Actually, our main product is acetyl CoA. This acetyl CoA is also required for the synthesis of fatty acids, right? To make the concept of these products clear, I will draw the structures so that it will be clear more. So now we are having structural explanation of beta oxidation of fatty acids. Here, first of all, we have the structure of palmitic acid. Palmitic acid consists of 16 carbon. The first carbon is there. The rest of the 12 carbon are these 12, 13, and then 14, 15, 16. So it is 16 carbon compound. Then the next we have the structure of palmitoyl CoA enzyme. To attach the CoA enzyme, we have to attach this group, CoA sulfur and then CoA, and the carbon with which functional group is attached is known as alpha carbon. So this carbon we have is alpha carbon, and the carbon next to alpha carbon is known as beta carbon. So here we have alpha carbon and here we have beta carbon. The next product we are supposed to make is alpha beta unsaturated palmitoyl CoA, which means that there will be unsaturation at alpha as well as beta carbon. Here we have removed their one of hydrogen and form a double bond. That's how they show unsaturation, and now this product named as alpha beta unsaturated palmitoyl CoA. The next compound we have beta hydroxy palmitoyl CoA, which means that there will be presence of hydroxyl group at beta carbon. So here we having beta carbon as it is alpha and it is beta carbon. At beta carbon, this OH hydroxyl group is attached, and now this compound is named as beta hydroxy palmitoyl CoA. Next step we have to form beta keto palmitoyl CoA, which means that keto group will be attached at beta carbon. So here we have this beta carbon as this one is alpha and this is beta. Keto group is here. This is the keto group present at beta carbon. Next we have to convert this beta keto palmitoyl CoA into myristoyl CoA and acetyl CoA. Myristoyl CoA is 14 carbon compound, and here we have myristoyl CoA. Here we have 12 carbon, 13 and 14, and then this acetyl CoA, that is 2 carbon product. Right, so that is the structural explanation of beta oxidation of fatty acids. Now we will see from one compound of palmitic acid or one molecule of palmitic acid how many acetyl coenzyme we get. So here palmitic acid converted into palmitoyl and ends into myristoyl CoA, that is 14 carbon compound and 1 acetyl CoA. This acetyl CoA will again form the fatty acid as it is the precursor of fatty acid formation, right? So then furthermore, this 14 carbon compound will convert it into 12 carbon compound, and we will again get another molecule of acetyl CoA. Okay, so let's see how many total acetyl coenzyme A we get. First of all, we get 14 carbon compound, that is myristoyl coenzyme A, and we get 1 acetyl coenzyme A here. Then this 14 carbon compound is converted into 12 carbon compound, and now the total acetyl coenzyme will be 2. Then this 12 carbon compound changes into 10, 8, 6, 4 and 2, and we get first acetyl coenzyme A, second acetyl coenzyme A, third one, fourth, fifth, sixth and 7. So here we are having 7 acetyl coenzyme A, but this last compound, that is 2 carbon, that is also acetyl coenzyme A. So total we get 8 acetyl coenzyme A. right? So we conclude that from one palmitic acid we get 8-acetyl coenzyme A. This 8-acetyl coenzyme A will again form one palmitic acid compound, and then this one palmitic acid compound will again break down into 8-acetyl coenzyme A. So this is a cycle which will be regulated by many feedback or feed-forward mechanisms, and it will continue to break and form. So that's all about for today's lecture about beta oxidation of fatty acids. If you want more lectures like this, let me know in the comment section.